So let's jump in. So this is the second of the series of the master class, the Cyclodextrin Masterclass that we are organizing. And today's event we dedicated to applications of cyclodextrins in biologics. So today we will not be dealing with small molecules, but applications in biologics. And this is the outline of the today's event. I'll share some very brief general considerations about cyclodextrins in case you don't know them, just a few slides, and then we'll deal with four main topics in the field: how they can be used with proteins, how they can be used with vaccines. There are some other biotech uses beyond the vaccines, mainly in the production of certain biotechnological products, and the uses in gene delivery. So let's see about general considerations. Very briefly, what are cyclodextrins? These are cyclic molecules that have a shape of a donut, as you can see in this blue figure, that have a cavity. The molecule itself is composed of glucose units. So it's a natural product, at least the unmodified cyclodextrin, made of starch enzymatically, and there are a lot of chemical modifications that can come to these OH groups of the cyclodextrin, which enable, of course, a wide variety of derivatives, as we will see today. So what they can do, the cavity itself is the key component, mostly, as you will see today. It will not be the only component, but the main component of the cyclodextrins uses, because it's hydrophobic. It can encapsulate certain hydrophobic molecules and change their properties apparently. So for a shorter amount of time, and these changes are exploited in different industries. Today, of course, we're mainly focusing on pharma, but they are there in foods, households, cosmetics, and a lot of industries. In the pharma, today we are dealing mainly with the novel applications. So currently the mainstream use is still small molecule formulation, solubilization, and chemical stabilization or taste masking mostly. What we'll talk about today is how they can be used in monoclonal antibody, gene therapy, and there are other applications which we will not talk about today, like how they can be used as active ingredients or in targeted therapies or in diagnostics or diagnostic field, maybe in another occasion. So what happens, this is what I said, that a hydrophobic compound enters the cyclodextrin cavity, and here you can see it's masked. So the original properties of the compound became, let's say, invisible for a certain amount of time, at least as long as it's in the cavity of cyclodextrin, and that can have a lot of interesting applications. Of course, with biologics, we'll see that these molecules are very big, so this interaction on the molecular level may not be necessarily as we see it here. So let's see how they work with proteins. With proteins, unlike with small molecules, the main application is not solubilization, although it can work in that area too. And here with proteins, what I mean in general is, okay, big proteins like molecular antibodies, but also smaller proteins, oligopeptides or polypeptides like insulin, as you can see, because they can interact with both. So with proteins, the primary application is prevention of aggregation, because the cyclodextrins can do that, can stabilize physically the protein and prevent their interactions. They can also help in reducing the viscosity, or at least not contributing to the viscosity, prevent the absorption of the protein on the wall of the container or the application surface, uh, or increase the solubility in certain cases. And this is just a visualization how this can be done. We'll go into case by case to see how these applications are manifested and achieved by the cyclodextrins. So stability of proteins is, of course, a very big issue, because proteins tend to aggregate, and they can also denaturate or suffer certain enzymatic degradation or oxidation. And this can be, of course, caused by different types of environmental stress or changes in the circumstances. Cyclodextrins were shown to be preventive to most of these applications. I'm not saying that in all cases cyclodextrins would stabilize your protein and prevent the degradation or the physical aggregation, but there is a lot of evidence saying that cyclodextrins can do that, and um more and more studies are emerging from these directions. Just to show you some practical examples with existing monoclonal antibodies and proteins, how this can be imagined. Um, in most cases, the cyclodextrins that are used in these cases are of course cyclodextrins that are approved for parenteral purposes, and that's not a very big number. So parenteral cyclodextrins or regulatory approved parenteral cyclodextrins means two different types only. One is the hydroxypropyl beta cyclodextrin, and the other one is the sulfabutyl beta cyclodextrin. Period. There is no other cyclodextrins that you can use in this field. So with proteins, these were the ones that were evaluated mostly. And since there is one cyclodextrin, the hydroxypropyl beta, which has a trade or a brand that's specifically proposed for used in biologics, the Kleptos Biopharma. This is where the most of the publicly available data is presented. So some of the results that I'm presenting, as you can see, comes from Rocket and their studies, because they have been doing a lot to make publicity for the application. But what you can see is that okay, the moda protein is here, the adalimumab, and as you can see in unstressed conditions, of course, there is a very limited particle concentration or aggregation, and upon stirring, the number of aggregates, obviously upon the interaction of the proteins, increases if they are kept in the control buffer. And here in this study, what is shown is very fascinating. So in the beginning, cyclodextrins were proposed as an alternative to polysorbates, so they could be a good uh, tool because their safety profile is much better than the polysorbates. Um, they're not metabolized, they're not degraded in the blood, no reactive oxygen species developing, and so on. So the original idea was to replace polysorbates with cyclodextrins, which is of course feasible. But what was discovered recently by colleagues at Rocket is that, as you can see, of course, polysorbate itself works very well for the stabilization of the protein, and so is cyclodextrin. Here you can see that there's a concentration difference. So usually for the stabilization, you have to use cyclodextrins in a higher amount compared to polysorbates. But the interesting thing is the synergistic effect of the two stabilizers that have a different mechanism of action. So as you can see, if you combine the two, there is a dramatic decrease in the aggregation and the particles formed. And this is something very fascinating. That's also the main innovation of some patents. So just bringing another example with some molecular modeling is the insulin, to show you that, of course, in this case with proteins, the size-wise comparison compared to small molecules is very different. Here we're not talking about one cyclodextrin interacting with one protein, much more like a surface effect. As you can see, several cyclodextrins attacking or interacting with the surface amino acids of a protein. And these interactions can be inclusion complex formation, like in the classical term, but they can also be uh, only secondary interactions with the surface of the cyclodextrin. So, of course, both the sulfabutyl ether beta cyclodextrin and the hydroxypropyl has different types of side chains capable of hydrogen bonding, or in case of the SBCD electrostatic interactions. So they would inherently react differently with the same protein. And of course, this also means that if such interactions are not taking place between a protein and the cyclodextrin, the stabilizing effect of the cyclodextrin will be much smaller, or in some cases, not present at all. Um, so if you look at the literature, you can see quite a few studies on different types of monoclonal antibodies, as well as other natural or designed proteins, and you can see a lot of examples. I'm not going into too much detail because it's not providing additional information. There are a lot of case studies showing that cyclodextrins can work in formulating proteins, and they are, of course, a very safe choice in excipients when formulating proteins. So it's a wise choice to be on the shelf if you develop formulations in this aspect. Just to bring you some other examples how cyclodextrins could in the future maybe stabilize proteins, is a different type of concept here, where we had the idea to create this self-assembly of cyclodextrins. So here you can see it's not a cyclodextrin polymer, but a series of cyclodextrins forming a chain-like structure that can cover the surface of the protein for the stabilizing effect. Uh, how the mechanism of action works is, of course, as mentioned, can be still based on both inclusion complex formation and surface interaction, or the combination of the two. And it would be very much dependent on the protein itself and on the tertiary structure of the protein itself. So how would cyclodextrins reduce the viscosity? This is more or less an inherent effect of the prevention of the aggregation, and it's also a concentration dependent effect of the cyclodextrin. So as you can see, the more cyclodextrin you put in the solution, the more interactions you would have with the protein, and that would lead less aggregation and eventually reducing the viscosity. One additional note on that. So the cyclodextrin itself, within this concentration range, so at fairly low concentrations, let's say up to 10-15% of cyclodextrin solution, has a viscosity very close to the media it's dissolved in. So it has an exponential increase in the viscosity, which only starts at the high cyclodextrin concentration. So in the range that they are applied in these formulations, they will not contribute themselves to the viscosity. The preventing of aggregation to the wall is also a combined effect, partially because cyclodextrins also cover the surface of the wall. They are, of course, present at a fairly high concentration in the solution, and they have, of course, the cyclodextrins. At least these cyclodextrins, what are applied for protein formulations, do not form aggregates themselves, so they do not interact with each other, and accordingly they would prevent the protein from uh, uh, absorbing to the wall. Despite the promising results, there are not too many cyclodextrins which are close to the market or on the market with protein or peptide formulations. So if talking about commercial formulations, I can only mention two. And of course, one is also a bit, questionable if we say that it's relevant to the topic. So it's Kyprolis is the blockbuster drug, which is nice, but it's not a classical protein. It's a tetrapeptide. So it's closer, maybe a bit to small molecule, and also the application is closer to a small molecule. So here the cyclodextrin is a solubilizer, but it shows that cyclodextrins can interact well with peptide structures. Beximi, which was more recently applied, product by Eli Lilly, is much closer. Of course, we could say it's a well fit in this field. It's a glucagon formulation as a intranasal formulation, and it's formulated with classical beta cyclodextrins. So, of course, in this case, we're not talking about parenteral administration. So, a wider selection of the cyclodextrins was possible, and this was the choice of Eli Lilly. That being said, there are no other commercial formulations, but quite a few products are in clinical or preclinical development. So I'm quite sure that these applications of cyclodextrins will be one of the main uses in the future. Just by one slide, I mentioned a topic that's slightly related. So there were some attempts to create monoclonal antibody cyclodextrin conjugates to improve the properties mainly of the antibodies with the help of the cyclodextrins. So this is something that is feasible to be done, and there are some studies of using such compounds, but it's not a very well spread application so far, but certainly a possibility. So let's see what are there in vaccines. In vaccines, there are much more commercial formulations using cyclodextrins, surprisingly or not surprisingly. Let's start with some historical introduction because the uses of cyclodextrins in vaccine has a surprisingly long history, almost as long as their use with small molecules. So Pfizer was the pioneer also in this case with the vaccines. And the first two examples that I'm about to show you are veterinary vaccines. So not human vaccines, but of course formulation-wise, conceptually, these do not differ much from human vaccines. So, Suvaxin was the very, very first product that contained cyclodextrin as a vaccine additive and not as a formulating agent, but as an adjuvant. And this contains a special cyclodextrin, which is special also by its structure and also by its application. So, this is a sulfolipocyclodextrin, as you can see here. So, it contains a longer aliphatic chain and sulfate groups, which are, of course, uh, related to the adjuvant effect of the product. And this product was developed in the 90s, so a long time ago. The peculiarity is that this was always the vaccine adjuvant that was used in this formulation, but there are no other products ever that contained the very same vaccine adjuvant, surprisingly. So this is a standalone application of this particular cyclodextrin. No one has any commercial manufacturing currently globally about this cyclodextrin, although it's clearly a potential vaccine adjuvant. So it's a very strange story, but it started opening the way to understand if there is a new use for cyclodextrins to be used them as vaccine adjuvants. The second product also contains the cyclodextrin as a vaccine adjuvant, but here the cyclodextrin is a common one. As you can see, it's the hydroxypropyl beta cyclodextrin. So the very same that was used to stabilize proteins and the very same that's in parenteral small molecule formulations. The vaccine is different. It's against Newcastle disease. Formulation-wise, it's not very special. Here, the peculiarity was that they also discovered that a very common parenteral excipient can be used as a vaccine adjuvant in some cases. And following up on this one, a much more recent study, and now we are going into human vaccines. So Daiichi Sankyo has an influenza vaccine in clinical studies, intranasal influenza vaccine, where again the cyclodextrin is used as vaccine adjuvant. And here I'm just showing you the comparative results of the original vaccine and the one where cyclodextrin is used as the adjuvant, and you can see uh, the incremental change in the T cell response, T cell related immune response that the presence of cyclodextrins triggered. Um, and so in this case, the vaccine itself, of course, has a local effect, and that's what was shown, that the cyclodextrins responded specifically, boosted specifically the T cell related immune response, and also they had some adherent effects like reducing the allergen risk of the vaccine, and also maintain a longer immune response. So this, of course, raises some questions: how the cyclodextrin is doing that, what is the mechanism of action, is that something that the cyclodextrins would do if we formulate small molecules with them, because obviously the dose is the same, more or less. In this case, well, with other commercial formulations. But it also opens a lot of doors. Okay, so now we have seen two different types of cyclodextrins that have an adjuvant effect. What else could be within the cyclodextrins that have an adjuvant effect, or could we design a cyclodextrin that has a really superior adjuvant effect? Because the sulfolipocyclodextrin was obviously developed for this purpose, but not used in other formulations, and hydroxypropyl beta cyclodextrin was different for a completely different purpose, and it was an accidental discovery that they revealed a potential adjuvant effect. A recent study that we did at Carbohyde, just to follow up on this, we screened a huge library of cyclodextrins and checked out how they could affect the activation of different monocytes and lymphocytes. We followed a lot of different biomarkers, and I'm not able to show you results in detail, just what's available now. So as you can see, the cyclodextrins that we tested are safe, and we use the negative and the positive control. So there's a mock formulation and E. coli lipopolysaccharide as a positive control for triggering the immune response. And as you can see, there are dramatic changes and dramatic differences between the different cyclodextrins. Some trigger immune response much beyond even the positive control, and while others still do, and of course we have seen a lot of cyclodextrins that trigger no immune response, uh, there are a lot of structure activity relationships that can be drawn here. So we're very positive, and we are actively working on creating a new generation of cyclodextrin that can be truly used as vaccine adjuvant. As you can see also from the different biomarkers, the response is very different depending on which type of cyclodextrin we use and what kind of immune response it triggers. So this was the vaccine adjuvant part. Of course, cyclodextrins can be used for other purposes in vaccines. I mean, other than vaccine adjuvant, and I'll bring a few examples for those. Obviously, if they are capable to stabilize proteins, it makes sense to assume that stabilizing vaccines or vaccine components or protein components of vaccines is something that cyclodextrins can also do, and that's the reality. So here's just an example of a patent and development from Sanofi Pasteur, who used the cyclodextrin in the formulation of a poliovirus vaccine, and it was shown that not only it stabilizes the the cyclodextrin, but it can also prevent it from the degradation which is caused by the preservative itself. So it's an interesting application of the cyclodextrin itself. A much more recent example for some events that we all remember. So one of the vaccines that were used in COVID's, Johnson & Johnson's vaccines, to be very more specific, contained cyclodextrin, again, the hydroxypropyl beta cyclodextrin that we talked about a lot today. So this is one of the components of the vaccine. And while it's not specifically mentioned from the patent, you can read that here the cyclodextrin is used not only as a stabilizer for for the components of the vaccine, but also as a cryopreservative. And using cyclodextrins in the cryopreservation process is something that has also been known for a long time. This application was mainly exploited in veterinary applications. But here, apparently, this is also something that can be used, and it's a potential application for cyclodextrins. So cryopreservation uh, is also a possibility. And of course, if you use cyclodextrins, all these how to say positive features of the compound come together. So the stabilizing and the cryopreservative effect will be simultaneously present in such a formulation. This is a bit more information if you want to go deeper into the cryoprotectant application. It's usually not the hydroxypropyl beta cyclodextrin, at least if you look at the literature. Okay, cyclodextrins and cryoprotection. Usually it would be the methylated cyclodextrin that are used, but these applications are typically um, not in vivo applications, so not in drug products, but more in research or in cell cultures and such like that. So you can see cryopreservation of T cells or maintenance of sperm and applications similar. But of course, this field also holds a lot of potential. And yeah, this is a bit more information about the Sanofi Pasteur patent that I mentioned. So cyclodextrins can also increase the chemical integrity of certain vaccine components. So these were the applications that are currently used in different type of vaccines. And now let's jump into other biotech uses. Although this will contain also some reference to vaccine applications. But here in this part, I would like to focus much more on how cyclodextrins could be used during the process, the biotechnological process. So productions that contain different kind of bacterial species, enzymes, and so on, and what are the possible uses of cyclodextrins. So this seem like research examples, but the majority of these applications are actually commercially used when such compounds are created. Uh, the application itself is not that different. I mean, the mechanism of action for the application is not that different from small molecules. It's just a different viewpoint how you take a look at them. So how you could improve the yield of a biotech formulation. There are technically two different approaches here. The first approach is that you use the cyclodextrins to solubilize the starting materials to enable them in higher concentration and make them available for the enzyme for the production. Or the other potential application is that often in these cases, the product of the process is an inhibitor of the reaction. And if the cyclodextrin encapsulates the product, it can mask its presence and accordingly give a boost to the production itself. And here you can see how um, different cyclodextrins improve the yield of the production because of the cholesterol complexation. Just another example where again different types of cyclodextrins were tried for the production of lancacidin. In this case, and the cyclodextrins were shown to have, of course, you can see the structural differences, and here the beta cyclodextrin was working best. It had very minimal effect on the microbial growth, and it was not consumed by the organisms, but the production here, as you can see, compared to the control, could be increased dramatically. Some cyclodextrins are often used to enable certain type of components or technically food for the microbia and help them in their in growing them. So here you can see that the naked cyclodextrin or the cyclodextrin complex of hexadecane had a dramatically different effect on the microbia growth. But the most interesting example is in the vaccine production is certainly a commercial example, and it has peculiarities from a lot of different aspects. So it's related to the production of pertussis toxin, and here you can see the concept of the production. And what the cyclodextrin does in this case, as I mentioned, so the byproducts of the process are inhibitors of the process. And what the cyclodextrin does, and as you can see, also different cyclodextrins were tested here, is that it complexes the final product. You can see that these fatty acids are ideal guests for a cyclodextrin complex. And then the enzyme do not recognizes these byproducts, these inhibitors, and that can boost the production process. And as you can see, this particular cyclodextrin named as DMAb was superior, and even at lower concentrations, it could increase dramatically the production process. Actually, the toxoid production was improved more than 100-fold just adding 1% of the cyclodextrin to the production media. Let's talk a little bit more about this cyclodextrin because it's a strange one. Of course, it's a methylated cyclodextrin that you may have heard of, but it's a very unique methylated cyclodextrin and rather rare as well. So comparing methylated cyclodextrins, not going into detail, this one is the methylated cyclodextrin that you probably all know. This is the one that's a component of a lot of cosmetics, also the one that's component of a lot of uh, household products and in the chemical industry, let's put it like that, and a very potent complex forming agent, also some pharmaceutical applications, and it's a compound that's randomly methylated. This compound, however, has a very specific substitution pattern. So, in its name, it's very similar to the other one in reality, and in its functional properties, it's very different, and it's also much more different to make, and accordingly much more expensive. But for this particular process, this particular vaccine production, its performance is way superior compared to the other methylated cyclodextrin. Unfortunately, so if you Google this cyclodextrin, this heptakis 2,6-DO-methyl beta cyclodextrin, you will find a lot of suppliers globally. And if you buy the product and if you analyze, you will realize that what you bought is usually this one. So making this product is has a very it's not easy and needs a very special knowledge and production process. And in the nomenclature, these two products are usually mixed with each other, and that leads to a lot of confusions in this field that seems very difficult to overcome. So this is the structure, and when I mentioned that it has a very particular substitution pattern, this is what I meant. So there are three OH groups, not going into too much chemistry, don't be afraid. Three OH groups on the cyclodextrin, and two of them, the 6 and the 2, are methylated, and the 3 is not methylated. At least that's the main compound, even though it's only a main component of the final product, but that's the concept of the compound. Whereas in a common methylated cyclodextrins, these methyl groups would be completely random distributed around the cyclodextrin. So if you really want to use this compound for pertussis vaccine or any other applications, finding a proper supplier that can supply a product that has the performance suitable would be very difficult and needs a very careful attention not to buy the other methylated cyclodextrin. So, last topic, gene delivery. Um, in advance, I must say that among the four applications, this is the less developed, although it has a lot of potential and a lot of great promise. But all the data that I'm about to share are from preclinical studies, so no cyclodextrin-based formulation ever entered. For gene delivery ever entered the clinical phase, but I don't really think that's too long to come. So, of course, just a very quick overview. What are the alternatives if we want to develop uh, a gene delivery formulation? Of course, the main challenge here, or the two main challenges, let's put it like that, when developing such a formulation, we have to protect the cargo, so the plasmid, the RNA, whatever it's the DNA, whatever it's put or we want to deliver. And we have to, once we could protect it, we have to deliver it successfully to the cells. And there are a lot of strategies around that, how this can be done. Some of the drug delivery systems that are in use or have been in use are listed here. And of course, there are viral and non-viral applications. Recently, also with the help, let's put it like that, the help of the COVID, the non-viral applications are getting more and more attention in this field, but there are a lot of challenges. And cyclodextrins were discovered to be a very good contributors, let's put it like that, to these kind of drug delivery systems as components. So not as the main element, but as part of the drug delivery systems, because people realize that, okay, into liposomes or different micelle-like structures, you can feed cyclodextrin, or you can combine them with the layer or put them on the surface and or graft them on the surface. And such applications of cyclodextrins are very useful. They usually come with a reduced immunogenicity, with an improved safety profile, and so on. So this is how the applications of cyclodextrins in gene delivery all started. And this is not what I want to talk about. What I want to talk about is how this developed to another generation where it was realized that cyclodextrins do not necessarily need a drug delivery system, I mean, an independent drug delivery system, to be able to deliver and transfer genes into cells. So four different type of concepts were created. I'm not going into detail to all of them, but you can create polymers of cyclodextrins, cationic polymers of cyclodextrins, like a polyimine, which can interact with the DNA. And I'm going to show you more information about this product because we have some nice collaborations with the Debrecen University about such product. You can make a polyrotexane, which, as you can see, is more like a necklace where the pearls are cyclodextrins, which can also interact with the DNA. You can create amphiphilic cyclodextrins, and this is the other field that I'll talk more about, where on one side of the cyclodextrins you put the aliphatic chains, on the other side of the cyclodextrins you put usually cationic groups. And in this case, technically the cavity doesn't play any role in the interaction. This cyclodextrin serves as a building block of a micelle-like structure, very similar to a lipid nanoparticle. Of course, with different properties, but conceptually, you get a nanoparticle, and in the inner cavity of the nanoparticle, you can encapsulate the gene. And this is the other one that I'll show a few more examples about, because this is the concept we are actively working in a European international consortium called GeneGut. And there is also another concept which is similar to what I've shown with the proteins, but here there are actually in vivo and in vitro reassuring data from the Sorbonne University that you can create self-assembling cyclodextrins that can surround an RNA thread or a DNA thread and protect it and deliver it to the cells. So these are not cyclodextrin polymers, just self-assembly of cyclodextrin monomers. So a few words about cyclodextrin polymers. This is something that's very interesting in the cyclodextrin area, and there are a lot of approaches how cyclodextrin polymers could be used. There is a very wide versatility what kind of polymers you can create. As you can see, you can create a lot of neutral and apolar types of polymers, cationic, anionic, depending on what kind of agent you use for the polymerization. It can be something that's biodegradable or non-biodegradable. So there's a huge versatility. And usually cyclodextrin polymers show very good complex forming abilities. So it's from a functional perspective, it's also a very nice tool. Unfortunately, not many of the applications have reached the market. So the only cyclodextrin polymer that's currently on the market that is not in the pharmaceutical field, but in the wastewater treatment field. But maybe that's something that we'll talk about at the next Cyclodextrin Masterclass. But anyhow, so cyclodextrin polymers can be made wide versatility. And what they would do, so here, this is a beta cyclodextrin polymer. It can be functionalized or complexed with uh, compounds containing positive charges, and this can bind the DNA itself. Of course, this modification can also be um, chemical modification. Here, it's a non-covalent modification, but it's a very strong binding to the DNA itself that can deliver it to the cells. So these kind of polyplexes have a lot of potential in the field, and we have also worked with them, but still we chose to go with the cyclodextrin monomers eventually. But they are certainly possible. So what we're working on is amphiphilic cyclodextrins, and of course, chemically, again, cyclodextrins are really versatile. So it's really up to the chemist's talent and consideration where to put the lipophilic chains, to put different types of same times, what kind of charged groups to put on the other end, and so on. So there's a huge versatility, and these amphiphilic cyclodextrins can create a nanoparticle and condense DNA in the middle. Some of the compounds that we have created, just okay, all the possible combinations of what you can see with a lot of different tracers, spacers. So it's a huge library we have created when we did the screening over 100 different types of compounds. And as I said, it was done in the gene gut consortium, not going into the details of the synthesis, rather showing some results of the cyclodextrins. So here we try to deliver an antisense oligonucleotide, and as you can see in the naked antisense oligonucleotide couldn't get into the cells, and we compared three different types of cyclodextrins to show how much of the cargo they can deliver into different cells. And of course, we will also tracking the protein levels in different in vitro models, how much they can to numerize the transfection uh, affinity. And as you can see, the structure of the cyclodextrin itself, even though these compounds are chemically rather similar, have a huge impact. Of course, depending on how you modify it, the size of the nanoparticle will be different. And the charge will be different, the encapsulation efficacy will be different, the transfection efficacy will be different. So we have worked a lot on understanding the structure-activity relationships of these compounds, but eventually what I can say is that such cyclodextrin nanoparticles seem to have a fair competitive edge or advantage compared to lipid nanoparticles for their very superior immunogenicity. So these are very safe compounds, and as far as we could tell so far, they're triggering a very, very limited or no immune response at all. And the transfection efficacy is comparable to lipid nanoparticles. So I'm not really saying that it's much better, but it's similar, let's say. And also the load, so the type of oligonucleotides that you can package within the cyclodextrin nanoparticles seem to have a very wide variability. So we were able to put smaller and larger oligonucleotides and deliver them successfully to different types of cells so far. So that's why I'm saying that this field certainly deserves attention. And not to say that, okay, it's not a topic of today's talk, but amphiphilic cyclodextrins have been known for a while. And of course, it's not necessarily only uh, oligonucleotides that can be encapsulated within the cyclodextrin cavity. So there are papers out there where small molecules are included within anti-cancer drugs and so on. So this kind of drug delivery system potential is not limited to gene delivery. Of course, it's a huge field, but it can be also used with small molecules or potentially other biologics as well. So, and I'm stopping talking very soon. So, as a takeaway message, I hope I could give you some new information about applications of cyclodextrins in the formulations of biologics. As you can see, there is plenty of potential applications and a lot of possibilities in all four fields. Uh, Vaccines are already there, and cyclodextrins are already used as vaccine adjuvants. So are they used to improve the productivity, usually, of certain biotech processes. The next upcoming big application, in my opinion, would be protein stabilization, and I'm 100% convinced it's just a matter of time when we will see the first cyclodextrin-based product on the market, simply because of the very favorable safety profile and the tremendous amount of safety data showing that these compounds are clearly suitable for parenteral administration. Gene delivery is something that's a bit further around, so it's not on the next corner, but maybe the subsequent one. And it will, of course, as you realize, delivering genes would need a new generation, chemically new generation of cyclodextrins. So in order to enable a platform like that, there is a lot of uh, GLP preclinical studies and a lot of clinical trials needed still. I'm rather sure that it's again just a matter of time and commitment when we will see uh, in the clinics at least some of these applications. So I really hope that you enjoyed the presentations, and let's see if we have any questions. So I'll stop sharing now. And yeah, hopefully I stop sharing. So now please drop your questions in the chat box. This is where I would be able to see them, and we'll have time to tackle a few of them. If there are more, feel free to send them to me via email or just put it in the chat box, and we'll try to answer those when we send the recordings of the webinar. Okay, yeah, I see Carmen's comment was completely right, mentioning that the HPBCD in the flu vaccine doesn't work as in a classical definition of immunological adjuvant. Yeah, that's completely right, Carmen. This kind of application of cyclodextrins is not the same as what we or how we think about vaccine adjuvants currently. And there is a lot of work to be done to explore what is the cyclodextrin's place in applications in vaccines. Unfortunately, there are not too many studies or too many systematic research that I know of who are dealing with that aspect. So to understand how cyclodextrins interact with an immune system, exactly how they influence the effect of vaccines, what kind of vaccines, what kind of immune response they could boost. This is something that's a gap in the public cyclodextrin knowledge, and there are no groups, to the best of my knowledge, who are actively working on that. So I really hope that maybe there could be some collaborations initiated around that, or maybe some nice European international collaborations to explore the effect or something like that. Yeah, thank you, Carmen, for the compliment. What would be, Julia asking, what would be the maximal size of proteins to be encapsulated by cyclodextrin? So, um, I don't really think there is a cap here because we are not talking about a classical encapsulation. So we are talking about ants covering an elephant, not ants eating the elephant. So uh, there are myriads of cyclodextrins. Okay, not myriads, but dozens of cyclodextrins interacting with the very same protein. If we have a bigger protein, there would be ideally or theoretically more cyclodextrins interacting with it, but that would be very much dependent on the protein itself. So with the protein, since it's a surface interaction, the size is not really a limiting factor. The hydrophobicity of the surface, uh, the potential of the surface to make uh, secondary interactions with the cyclodextrins, those would be the limiting factors of the success or unsuccess of the formulation. The size of the protein doesn't really matter. Simone is asking, are there documented cases of CD delivery systems for metal drugs such as cisplatin or carboplatin? Uh, well, this is something that I don't know by heart. I would be uh, very surprised. So finding compounds that are not how to say, finding compounds that completely lack any evidence of cyclodextrin interaction is very rare. I haven't worked specifically with cisplatin or carboplatin, so I admit now that okay, I just Googled and there is a paper claiming that okay, I don't know, cisplatin cyclodextrin complexes are to be used in anti-cancer agents. So as far as the structure of the compound would allow, it would make sense. So for cisplatin, there is certainly evidence that this is something could be done. I don't have personal experience with such metal drugs. Bill is asking, hi Bill, would a mixture of cyclodextrins be useful in stabilizing proteins? That's a very good question, and I think it is. So it has relevance. It's very rare in the pharmaceutical development that different types of cyclodextrins are used in the same formulation, but it's not without example. So with the small molecules, there is at least one commercial drug which contains two different types of cyclodextrins. But of course, with small molecules, since it's usually a one-to-one interaction, it doesn't usually it doesn't make sense to combine multiple cyclodextrins. With proteins, there is it's a very good point because with proteins it could make sense as the protein is huge, the surface is huge, and there could be proteins that interact Okay, now we're talking about the sulfobutyl cyclodextrin making electrostatic interactions more. And there could be areas which like the neutral cyclodextrin for hydrogen bonding more. So I haven't seen any papers dealing with a concept like that. Maybe Carver knows if they are, but theoretically it would make sense, and it's possible that it could work very well with some proteins. Probably not with all proteins, but it's a very nice research area. So yeah, where does cyclodextrins release the drug into the body? Gerhard asking. So the cyclodextrin drug interaction is fairly weak, talking about chemical terms. So we're not talking about a covalent interaction. It's just a temporary interaction, and it's very weak. So if we are talking about proteins, the cyclodextrin would release the protein immediately. So if you administer it intravenously, the millisecond the cyclodextrin complex, okay, the cyclodextrin formulation enters the bloodstream, and this is true with proteins. This is true with small molecules. Most cases I'm talking about, there are exceptions. The there is a huge dilution, and the cyclodextrins would look for other guests within the body which are more appropriate, and the protein would also look for other parties to interact with. So the complex or this associate wouldn't exist anymore. That's why we're always saying that when you stabilize a protein, uh, the protein stabilization can work in the vial. It's a physical stabilization, but a cyclodextrin, no matter how well it stabilizes the protein in the vial, it will never be able to protect it from the enzymes on its own, because this this structure, cyclodextrins covering the protein, doesn't exist in the body anymore after you administer. Of course, if you're talking about subcutaneous administration, the dilution factor is much more different than with an intravenous one. So there, the release might be a bit slower, but conceptually, it will be the same. So it will release it where you administer it to the body. With the gene delivery, things would be different, because here, there, the of course it's a nanoparticle. So in that case, the target is to be released only within the cells. Um, okay, Carmen helping me out with the answer. Thanks a lot, Michael Lynch. Share the paper, cyclodextrin-based delivery system for in vitro anticancer therapies. Yes, for Simona. Yeah, thank you very much for helping me out, Michael. So, Bill has a question. Bill's questions I already answered just now. So our time is up. I'm sorry, I was a bit too loose mouth, and we didn't have too much time for questions. But if you have any further questions about the topic, okay, well, let's tackle Michael's questions. Is there any evidence for cyclodextrins to facilitate the passage of therapeutic molecules across the blood-brain barrier? So, Michael, yes, there is such evidence, but it's not as straightforward as you might think, and I don't really think that it's relevant for biologics, but for small molecules, there is evidence like that. Uh, that if you administer the active ingredient without cyclodextrin, or compared to administering the active ingredient in a cyclodextrin formulation, the cyclodextrin-based formulation has a higher penetration to the blood-brain barrier. But it's not because there is a complex that travels in the bloodstream all through your brain and sits on the blood-brain barrier, where the cyclodextrin opens the or loosens up the blood-brain barrier and helps the molecule enter. It's mainly because the cyclodextrin-based formulation enables a higher blood level of the active ingredient, and accordingly, a higher portion can get through the blood-brain barrier. So cyclodextrins can be used as permeation enhancers in some cases, but for the blood-brain barrier itself, we cannot talk about this kind of permeation enhancing effect. It's simply, or mostly, I would say, it's the solubilization uh, uh, contribution and the higher blood level that's leading to an increased CNS permeation. There are attempts to design a drug delivery system that can actually deliver something specifically and targetedly to the brain, but that's beyond the current topic. I can share you some literatures on that if you drop me an email and would like to learn more about this one. Nose to brain delivery using cyclodextrins to cross the blood-brain barrier. Okay, Teresa, so uh, that's something that's possible. So, but it's a different concept because, okay, so for Michael, I was talking about if you deliver something parenterally, this is how cyclodextrins would react. If you deliver something intranasally, the effect of cyclodextrins is different because in that case, you administer the cyclodextrin formulation to the nose, and there is a high local concentration of the cyclodextrin, and there is no immediate dilution, like I mentioned, so the release of the complex doesn't happen. There, in that case, okay, use the nasal spray, let's put it like that, and the cyclodextrin complex is more or less intact in the surface of your um, nasal membrane. And some cyclodextrins, not all of them, but some cyclodextrins can interact with membrane proteins and extract cholesterol or fatty acid from cell membranes, which leads to a temporary loosening up of the membrane, and that can increase the permeation of certain drugs. For topical applications, this is something that can reasonably happen. So when you use a cream with a cyclodextrin, this is something, and the proper cyclodextrin, this is something that can happen. And for intranasal application, we have a lot of evidence that this is actually what happens. Cyclodextrin loosens up the membrane and helps the molecule permeate, and in this case, potentially get to the brain or even the cyclodextrin itself. So you're right, in this particular application, it's possible to improve the penetration. Yeah, nose to brain route. In combination of mucoadhesive, yeah, that's even more wonderful application. So yeah, if you combine it with a mucoadhesive cyclodextrin, which also exists, uh, then in that case, of course, the effect is longer and can help even more. So that's certainly a very valid therapeutic application, although I would say more for small molecules, but I don't really think that anyone tried with proteins or biologics. But anyhow, it's feasible. So, but still, our time is up. So if you have any further questions, I'm really happy to answer anything. Just drop me an email. Uh, and yeah, we can continue discussion. So again, I hope you enjoyed today's session, and we plan to continue, and we'll think what to do in late February, probably, but you'll receive notification if you follow us on social media or if you are subscribed to our newsletters. Thank you for your attention. It was nice to talk with you. Let's put it like that. Take care, guys. Bye bye.